So I'm under strict orders to keep this to 15 minutes. So what I will tell you to do is I feel like an interloper here, because I suspect that cancer is not a common topic amongst all of you. So as you can see from the title, my attempt is to point out to you that cancer and development, in my opinion, are two sides of the same coin. So to that end, let me just also sort of set a stage, hopefully. I'd like to get you to think a little bit differently about, your Mobile 101 background. I mean, most of you have all been told that, the world revolves around DNA, and DNA nicely makes messenger RNA, and it makes protein, and life is simple and good. I would submit to you that's not true. That in fact, if you look historically, the first, biologic molecules on this planet were almost certainly RNA, not DNA. And RNA is a whole lot older, a whole lot more complex, and it probably actually regulates almost everything, including DNA and protein, by the way. So the real world is that RNA, I think, is at the pinnacle of this whole story. And then you're going to say, yeah, nice theory, but what's the proof of that? So the next several slides, what I'd like to do is try to convince you that there is actually a compelling body of evidence to argue strongly that RNA in general, and particularly non-coding RNA, is in fact a dominant biologic factor in all sorts of functions, including development, regeneration, and cancer, by the way. So where does that information come from? The paper that we published about a year ago specifically pointing out that the majority of RNA in any given cell is non-coding RNA. And if you think about it for a moment, Mother Nature's not stupid. Why would she make so much non-coding RNA if there wasn't a reason? And I think that's the first clue that this is not just transcriptional noise, as a few unenlightened people have suggested, because there's a really, I think, strong body of evidence already extant and more every day that would argue that, in fact, this stuff is very, very functional. So what's the evidence for that? This is just a few papers from the literature. You can read as well as I can. There's almost every year, the number of such papers is doubling. Certainly it's increasing every year dramatically as more and more functions for non-coding RNA are identified. So one I found particularly interesting actually is a paper, trends in I think medicine, if I'm not mistaken, molecular medicine. The neurobiologists actually have done us a favor, pointing out that these non-coding RNAs control almost every aspect of neurodevelopment and neurocognition. And I think, when you have something that is this pervasive and with this many players as listed here, it's unlikely that this is transcriptional noise. So in our particular case, I have to apologize, as I told you, we happen to do cancer, sorry, but that's just development gone awry, as you know. So we actually focused on a number of things. One of our favorites happens to be Ewing sarcoma. And James Ewing described this tumor back in 1921. I had no idea what it was because it was completely undifferentiated, as the lower right-hand panel would indicate. It's probably the most undifferentiated of all human malignancies. So it's always been an enigma as to quote what it is. And the answer is probably that what it is a function of non-coding RNA. To set the stage, I'm sorry the dots are so small you can't see this, but I want to just explain that if you look at transcripts in cancers, on the left hand lymphoma, the right hand neuroblastoma, both of which are common malignancies, particularly in children, and below that in the black panel are a listing of coding in red and non-coding in blue. And it's roughly 50-50 for these two relatively differentiated malignancies. So that these are the dominant transcripts in these tumors, and it's about a 50-50 ratio of coding versus non-coding RNA. And this is by significance of association with the entity itself versus all other malignancies. If you go a little bit farther and look at Ewing's, for example now, it turns out that the blue are non-coding RNA transcripts, the red are coding genes, and as you can see right away, there's far more non-coding than coding. And secondly, this is a plot of magnitude of association on the y-axis and significance, statistical significance on the x-axis. And it's pretty obvious that the entity in the upper right-hand corner there is the most abundant and most significant transcript for Ewing sarcoma, and it's non-coding. The next closest coding gene is listed down below, and that's an order of magnitude less abundant and less significant. So clearly, there must be a reason for all this. And to put this in perspective, if you look across the landscape of cancer and ask, where's this thing expressed? And the left-hand side is this is a log base 2 y-axis, by the way. Ewing's is listed on the extreme left, and then all the other common players in the field of cancer are listed to the right. And nothing comes close to the levels of expression of this transcript found in Ewing's. So it's highly tumor specific, okay? But what's more interesting, I think from this audience perspective, is where is it expressed in normal tissue? And we've looked at all sorts of normal tissues, which I don't include here. It's not expressed in mature normal tissue. And we looked at developing tissues, and in particular in brain. Fortunately, there's a project underway at USC right now, which Jim Knowles is doing, looking at developing brain at different stages in different regions. And we had the data set available, we looked, and it turns out that this same transcript is expressed in the second trimester developing corpus striatum, and to a lesser extent in the temporalis and thalamus regions. So it's very specific in its spatiotemporal expression, and also happens to be in Ewing's, as I told you. Also, you might ask, what does it do? What's the evidence that does anything functional? I won't go into the details, but I'll just point out to you that this thing is involved in the polycomb repressor complex, including the trithorax and PRC2 complex that, as you know, controls epigenetic remodeling across the genome. And this is by no means the only such transcript. We find other transcripts in these tumors that are completely novel and strange. Another one, for example, this is looking at primary Ewing's versus metastatic Ewing's. The upper bar shows a 400 kb continuous RNA transcript expressed in the primary and reduced expression in the metastasis. This is a completely intergenic area. Conversely, we find other non-coding RNAs upregulated in the metastasis compared to the primary. So it can go either way, but there's a consistent pattern of association. And I will note that metastases typically are less differentiated than primaries, i.e. more stem cell-like, as you can imagine. The other thing we're finding, and this is a little bit complicated, but bear with me, on the right-hand column you see primary and metastasis. And it's a heat map of relative level of expression of one non-coding RNA, the one I alluded to a moment ago. And in the primary, that sort of magenta box indicates that essentially almost all of the isoforms of this non-coding RNA transcript expressed in the primary tumor is of one type. But once you develop the metastasis, the second column, there is no dominant species to speak of. In other words, there's a loss of splicing control and expression of a non-coding RNA with the development of metastasis. So I think, and then you can say, well, all right, this is all interesting biology, but what relevance does this have to a real biologic parameter? Well, how about prognosis? I mean, certainly, 100 patients walk in the door. How do you know which ones are going to live and which ones are not? We've done a lot of stuff looking at prognostic profiles using so-called metagenes, an agglomeration of multiple genes. This is a 24 non-coding RNA prognostic profile, and this is a group of patients for whom there's no evidence that one will do better than another. No one has ever been able to figure out who are the ones that are going to survive and which ones are not. And as you can see, you can very easily determine who's going to survive and who's not using just 24 non-coding RNA transcripts. This is the same analysis using coding RNA. So I think you can appreciate a little better predictor with non-coding RNA than with coding RNA. So clearly, if these were random events, this would not be possible. The other thing I want to point out to you is that this particular transcript I showed earlier is actually surrounded by the red box. And also in the same locus is a known protein encoding gene called FEZF1, which is bounded by the blue box in the upper left-hand side. It turns out that they share the first exon. And one of the questions that comes up is, what's the relationship between a non-coding RNA and a coding RNA that share the same first exon on opposite strands of the DNA? And the answer is that, first of all, this same coding gene, FEZF1, which as of a year ago had exactly one reference in the world's literature, is a transcription factor expressed in developing brain in corpus striatum, temporalis, and thalamus. The exact same expression pattern as the non-coding RNA that we showed you a moment ago. The other thing is, if you look at co-expression analysis, it's almost perfect co-expression of the two. So both of them are expressed in developing brain. It turns out they're slightly different in time frame, but in point of fact, they are co-expressed, which leads to the thought, what's the relationship between the regulatory elements of the non-coding RNA and the coding RNA? And there's increasing evidence that, in fact, these things tend to get co-expressed under as yet unknown regulatory elements, whereby you get a coding and a non-coding expressed off the same locus. Obviously, one of them makes protein and the other is almost certainly regulatory. So what's the evidence for this? Well, there's actually beautiful evidence in the field of developmental biology. And just a couple examples, the BXDL gene, which is a long non-coding RNA, binds to the UBX promoter and actually controls expression of this so-called ultrabithorax promoter region. and basically recruits ASH1, which is a trithorax response element, you know, the reverse of polycomb repressor. This is, if you will, promotes expression as opposed to suppresses it with polycomb. And so clearly, if you look at the graph, you see that there's almost exact coordinate expression of the non-coding RNA and the coding RNA. And this is true particularly of this EVX1, which seems to control the HOX-B5 6A cluster. This is an anti-transcript that comes off, antisense transcript that comes off the same locus as the HOX5B6 locus. And it appears that in fact they're co-expressed, as you can see here, in these developing embryos. And again, you have an example of a feedback loop, it seems, between a non-coding RNA regulating the expression of a coding RNA. And the theme, I think, that you see from all of this is that these non-coding RNAs are highly functional. They can regulate many processes. They're obviously highly expressed and active in predicting things like outcome and even certain functions in cancer. And as I said, cancer is best viewed as aberrant, if you will, development. And yet again, you see the same theme in simple developmental systems like this. And with that, hopefully it was at 15 minutes, give or take a little bit. And I think actually I made it, didn't I, David?
30 seconds to spare.
Thank you, thank you.
Yeah. Is it, maybe it's just epiphenomenon. What happens if you knock out this non-coding RNA? Can you do that?
Yes, and lots of bad things happen, or good things, depending on the setting. There's a number of studies. The classic, in my opinion, is something called hot air, which is a HOX locus antisense transcript. It's been shown, for example, that with elevated expression of hot air in women who have breast cancer, it progresses from a locally contained breast cancer to a highly invasive and metastatic breast cancer. And it appears that that is mediated by the so-called epithelial mesenchymal transition, which hot air and its 300 target genes, by the way, mediate, such that instead of having a contained breast cancer inside the basal membrane, it obviously invades through the basal membrane, develops mesenchymal phenotype, metastasizes, and is ultimately fatal. And so it's very clear, it's been shown that this is strongly associated, and certain studies done by that group, John Rinn at the Broad at MIT, have shown that in fact you can modulate the relative aggressiveness of breast cancer, for example. And there are now many, many other examples like this coming out. But I think you're going to see more and more of this in the field. These things are highly functional, and some work that we've done in Chichel Physiology, who's here, has been working on, shows a very similar phenomenon in this tumor system.
So is there commonality in the targets between the coding and non-coding sequences that are expressed together?
It'd be premature for me to make a...
the same regulatory coin, or they just happen to be in the same locus?
I think that there's a likely theme. I mean, the fact that, for example, hot air is an antisense transcript off of the homeobox genes, controlling epithelial differentiation, among other things. I think the reason these things tend to coexist is that, over eons, presumably, they have evolved functions that are complementary to one another. But they're not all just local phenomena either. Hot air is known to have at least 300 downstream target genes. So there's obviously a local phenomenon and, if you will, a distant phenomenon whereby this one transcript can both control expression in the HOX locus, but also a number of other genes as well. The theme that actually is emerging, which is kind of an interesting one, is polycomb repressor complex and also trithorax, pro and con for expression and epigenetic remodeling. Why do those things settle down where they settle? And it's more and more becoming obvious that the polycomb repressor complex or trithorax complex is guided to the promoter regions, either activated or suppressed by a guide RNA, and the guide RNA is a long non-coding RNA.
Cool. Yes, yeah. What's known about the difference in the expression of these non-coding between malignant and non-malignant transcripts?
It appears that the more differentiated the malignancy, the less they play a role. And the more undifferentiated stem cell-like, the greater the percent expression of these non-coding RNAs compared to coding RNAs. The balance shifts, the more primitive the tumor, to a more stem cell-like character. And it's known that non-coding RNAs in general are expressed at their highest levels during development, and they decline with maturation of the tissue and the organism. So I think what you're seeing is more aggressive tumors are more stem cell-like, and they also express more non-coding RNAs.
And what about tissue repair? What about injury repair? Are they expressed at a higher level at that point?
I don't know. I've never looked. It's a good question.
How many of these non-coding RNAs have enzymatic activity in and of themselves, as opposed to just regulating the expression of coding RNAs?
Now you're into an area that I was hoping to avoid tonight. Thank you very much.
I mean, you alluded to it yourself at the beginning, talking about the RNA origins of life.
Yeah. So I think that's actually the real issue. The ones that I'm talking about right now are, I think, ones that are probably regulatory elements in complex processes like, say, development and cancer and so forth. But there are vast numbers of other non-coding RNAs with very discrete functions. The scary thing right now is that I don't think anyone, first of all, has a clue how many there are, because let's start with the fact that the coding genome is roughly 1% of 3.3 billion bases in the genome. The non-coding is all the rest. And they tend to be expressed at lower levels, but they have far greater complexity. And the obvious question is, if 20,000 proteins, protein coding genes constitute what's standing in front of you right now, for example, or maybe there, the reality is, the truth of the matter is, we have no clue what the vast majority of these non-coding RNAs are doing. But if you think back, as I suggested, sort of, in antediluvian times, these had functions including a substitute for enzymatic activity that you would otherwise have in proteins. And I think what you're going to find is that they are almost certainly enzymatically controlling both nucleic acid remodeling and splicing and among a number of other functions, just like the spliceosome, thank you, and by the way, the ribosome. And you're also going to find that it probably has important effects on termination of message or survival of message RNA. So the number of functions is going to be, I think, profound. Some of these are going to protect RNA from enzymatic degradation, others are going to degrade RNA. And so, the repertoire of functions, I think we're just scratching the surface of right now, and I can't even conceive today of how many different functions. Well, what we do know, people like Tom Keck pointed out many years ago, RNA can do pretty much everything that any protein can.
So I think this is a good point to stop. I think it points out that we actually, Thomas Edison was vastly underestimating the complexity of the issue, and it's not 1/billionth of 1% of what we need to know. It's probably a billion billionths of 1% of what we need to know.
23 maybe, yes.
Yeah. So thank you for that, Tim. That was just great.
Did I get dinner too?
Yeah.
Oh, good. Thank you. All right.